The second subsession of the Plant Sciences session. I'm Ellen Dean. I'm co-chair of the Plant Sciences session. My co-chair, Sylvia Holtain, is in charge of the subsessions tomorrow. And we're going to start this subsession with Brian Anacker, who is currently at UC Davis and soon to be living in Colorado. Yay, Brian. And he's going to be talking about phylogenetic relatedness predicts ecological similarity, evidence from a 2-year field experiment.
Thank you. It's nice to see some familiar faces in the audience. Thanks for coming. Okay, how's this? Okay, I know you, Frank. I think we're all aware that phylogeny contains a lot of information about species differences. If we look at this painting here by Oscar Clarke, which depicts the evolutionary relationships among angiosperms, we can look at the colored branches and some of the symbols. And what those are indications of traits that represent shared derived traits, which are essential for the systematist to put names on species and to group species into higher taxa, like genera and families. And recently, there's been an interest in by ecologists in phylogeny, asking the question, can we use phylogeny to help us predict whether or not two species can coexist? All of our theories of species coexistence, or nearly all of them, are based on some level of species differences. And so can we use phylogeny to add a predictive understanding of the outcomes of species interactions on a local scale, like that here in this plot at Bodega Bay? So what are the predictions we might make from phylogeny for species coexistence? Well, there are two different categories. The first is called environmental filtering. So the idea here is that across the gradient of phylogenetic distance that separates species pairs, on the x-axis, we can say something about the probability of coexistence. And the first prediction here is that the probability of coexistence will decline with increasing phylogenetic distance. So if we take the case of close relatives, they're going to have a lot of the same traits and perhaps share similar niche preferences. And so they're going to be filtered into similar habitats. So we should expect them to coexist quite regularly. More distantly related species have had more time to diverge in their traits and might use different resources. And so they might be filtered into really different habitats on the landscape. And so we fundamentally don't see them co-occurring, and they may not be able to coexist. We can make the exact opposite kind of prediction given species interactions. Here, close relatives use a lot of the same niches, and so if they are placed together in the same plot, they're going to be going after the same resources. And if two ecologically identical or highly similar species are put into competition with one another, usually both of them can't persist in that site over long periods of time. So the coexistence expectation for close relatives given species interactions might be that it would be quite low. More distantly related species here could possibly facilitate each other if they have diverged in their niche preferences. So here we can think of a nurse plant species that might facilitate an herbaceous ruderal plant species in its understory. So we want to address these questions in an experimental framework. The first, does the ecological similarity of plants decrease with the relatedness? I think that's a very open question. And second, what is the balance of environmental filtering and competition in terms of crosswalking the relatedness of two species to their probability of coexistence? And if we find a signal for phylogeny in predicting co-occurrence and local plots, what are the underlying niche aspects? Can we measure those and find correlations between phylogenetic distance and niche distance, or the differences in two species in terms of what they do? So we conducted an experimental transplant study at the Bodega Bay Marine Reserve, which is a UC natural reserve. It's a relatively small reserve on the coast of Sonoma County, north of here. just over 360 acres. I classify them into 5 main terrestrial habitats. The predominant habitat is coastal prairie. The second most common is sand dune, and then this is the road that you take to get out to the marine reserve right here. It's also where the San Andreas Fault runs, and so that's a main border separating out the prairie from the dune. But then you have these other habitats, the bluff habitats along the edge, and then freshwater wetlands, and then there's also a little bit of chaparral type habitat, shrubland. In total, there's 466 vascular plant species identified here, predominantly native, so there's 295. Another unique thing is that in many parts of the reserve, the abundance of any of the plants in a particular plot is predominantly native. There's definitely exceptions to that in some areas, but it's a nice place for us because of the diversity of habitat types in a relatively small area and due to the abundance of native plant species and cover. So what we did was to do an experimental transplant where, for example, we take a focal plant species, here's Plantago erecta, and we transplant it into 4 different sites, spanning a range of phylogenetic distance from really low to quite high, so something around 0 when you're getting transplanted into the sites of conspecific plants, something around 5 to 25 million years when you're transplanted into the sites of congeneric plants, so here's Plantago subnuda, and where it grows at the reserve, something 30 to 55 million years here when you're getting transplanted into the site of conchlomelioles. Here's Nebulanthus texanus growing in sand dune. And then finally into the site of what we're calling a distant relative. So that's anything above the conchlomelio designation, so above that 55 million year benchmark. And those are just dates I'm throwing out based on the phylogeny we created, which is imperfect, and there's error around those dates, but just to say that there's some spectrum of phylogenetic distance that we're targeting. We also know that within these plots, it's not that Plantago subnuda is the only plant species there. So while we're measuring one phylogenetic distance, which separates Plantago erecta from Plantago subnuda, we also acknowledge that there's other species in the background community. But we try to pick sites where Plantago subnuda, for example, is quite common. So it's a simplification of the relatedness of our focal plant to the recipient microsite, but with that in mind, we'll move forward. And then within each place where we put our experimental transplants, we are going to reduce the neighbors. So what we're trying to do here is assess the degree of interspecific competition. And so here's a case where neighbors are left at ambient condition, and here's a case where neighbors are reduced. And we use clipping to reduce these, and we don't do a complete eradication because we don't want to have as many, or we want to minimize the unintended consequences of doing clearing, which includes drying out the soils and making a really abiotically stressful environment. But we do want to manipulate the presence of plant competitors. So for example, here's Plantago erecta in a bed box, and then we plant it into its congener's microsite, Plantago subnuda. Then we plant it into its conchlomelio site, Nebulanthus, formerly Linaria, and then Trifolium ficcatum, which is in Fabaceae. So those are the 4 places we put Plantago erecta, and then we repeat that for 14 additional species, each of them going into one of these 4 types of sites. We replace those taxonomic ranks with the phylogenetic distance using this phylogenetic tree we created. We do the neighbor reduction, which has 2 levels, control or ambient and reduction. And then we do 2 replications. And then within each little plot, we put 5 to 10 transplants, depending on the availability of plants. And the idea there was that we expected some level of mortality just due to transplant stress, but also perhaps as a response to the environment. So this allows us to estimate survival in each plot. And then we did the really foolish thing of conducting this over 2 years. Not every species was planted in both years. I think 8 of the species were present in both, and then we used a unique subset in year 1. That did really poorly, so we didn't use those again, at least a different set for year 2. I'm not going to really talk about the year effects. They were really interesting. I'm going to see if phylogenetic distance and removal have clear patterns despite these kind of year effects. So we're just going to treat that as a nuisance parameter. So talking about the analysis, we're going to fit one of these linear mixed effects models. We're going to try to predict the final biomass of our transplants at the end of the growing season using their parameters of interest. So we call these our fixed effects the phylogenetic distance separating a focal plant to the recipient microsite as it interacts with neighbor reduction. And then we've got these random effects. Here I mentioned species, so we know there's going to be a lot of contingencies by species, and we could talk about that until we're blue in the face, but what would be really interesting is if these parameters were strong predictors of final biomass after controlling for species. So that's what I'm going to show you today. And then site, that's just a geographic location that says that the removal treatment is nested spatially, so that's just another thing to control for. And then I'm just going to show you the biomass results. And like I mentioned before, we also have survival. They actually showed pretty similar results. We could talk about those if you're interested. So the questions we want to address here are, does the ecological similarity of any pair of plants decrease with their phylogenetic relatedness? And we're not going to use the conventional approach where we're going to measure the traits of these plants. We're going to actually use this transplant experiment to tell us where the plant likes to grow, and hopefully that reflects something about its niche preferences. We're using the plant kind of as a thermometer. We don't really know what the niches are per se with this part of the study. We just want to see how the plant does when we put it in places and see what happens. And then what is the balance of this environmental filtering and competition and response of our plants when we put it into these different microsites? And then secondly, what are the niche aspects that may underlie the phylogenetic relatedness effect on plant performance, if any? So then we're going to go ahead and measure some aspects of the environment and see how they correlate with the phylogenetic distance that I've described. So we just first started by looking at plots of the raw data, and we saw evidence for clear nonlinearities. So if we look at these 4 species here, we see Plantago erecta, when I mentioned a couple times, its congenitor, which we also used as a focal, Plantago subnuda, Rumex occidentalis, and Senicula crassicaulis. So what you can see is as you go out with phylogenetic distance on the x-axis, plant performance declines from conspecific through congenitor to confamilial, but then it goes back up and becomes more variable. So we saw that in these 4 cases and a bunch of others, so we tested right away to see if overall we have evidence for a nonlinear relationship between these two, and indeed we do. So I'm just going to show you this briefly. This is all the data. So 15 different species in the control plots and the neighbor reduction plots. And then I fit a nonlinear fixed effects line here with confidence intervals given the uncertainty in the data. And these are the statistics that describe that fit. So I just wanted to show you all the data to say that if you look at the raw data with all species combined, it's really tough to see what the heck's going on. And so that's where we're going to use our fixed effects model to come to the rescue and assign all the different species their own intercepts, assign different intercepts for the years, and take care of that nested structure in the design. So I'm going to take away that data and just show you the fits, ignoring all that noise. And this is what the model says. With increasing phylogenetic distance, there's a decline in performance up to some threshold, whereupon it increases again. Not only does it increase, but it becomes more variable, so you can see this kind of fattening of the tail on the right side of these plots. So that's a significant fit of the polynomial to the data. And it says there's a phylogenetic threshold, and I've just drawn a line here which indicates 63 million years, which is the maximum age of any of our confamilials. So that's a phylogenetic breakpoint. So below that, so below the family level, there's a relationship where there's declining performance with increasing phylogenetic distance. So to look at that a little more succinctly, I'm going to subset away all these distant relative sites and look at the data one more time. So what you can see here is a strong effect of phylogenetic distance. Now I'm just spitting it as a linear term, so there's no more curvature, because I got rid of all this data out here that forces it to go back up. There's a clear effect of neighbor reduction, where performance is higher when we take away the neighbors. That's exactly what we'd expect if there's competition suppressing plant growth. And then here we have a significant interaction, which we didn't see before. Below the family level, we can see a clear significant interaction where the pattern is much stronger in the absence of plant competitors. So both of these suggest that environmental filtering is happening, such that you do better in the sites of your close relatives and you do worse in the sites of your confamilials. You see that with and without the presence of neighbors. So it says there's something about the environment, either an abiotic aspect of the soils or perhaps a biotic component of the soil or some other above ground abiotic factor that is correlated with phylogenetic distance perhaps, which says that my niche if I'm growing in the site of my close relative, that's a niche I like, I'm going to do well there. The other thing we can see here is there's an indication for more competition in the site of close relatives. So here among close relatives, when we remove neighbors, there's a big effect, so a big release from competition, and that release is minimized in the sites of confamilials. So there's environmental filtering and competition present in these results. Because both of these lines are negative, that is, in the presence of competitors, we still see the signal of environmental filtering, a negative relationship. I think we can conclude that environmental filtering is somewhat stronger than competition. I'll leave it up to you to decide. So I think what's going on here is we've got these antagonistic forces going on when we put Plantago erecta into its own sites. Those are sites, and its site of its congeners, those are sites it really likes, but it also has to deal with strong competition in those places. Competition is less of an issue when you get out here, but then you're growing in a sand dune, which is really quite different from the conditions you want to be in, so performance goes down markedly. So signal is really lost above the family level, so there's a phylogenetic threshold there, or taxonomic if you will, and then competition is stronger among close relatives. So can we pick out which niche axes are reflected in this phylogenetic distance pattern? Well, what we tried to do here was measure 3 aspects that we thought should be important to plant growth. First is plant productivity, which should just be a correlate of the amount of stress in the environment. This is just the total amount of above ground biomass in a ½ meter squared plot. So we clip the biomass, dry it down, and weigh it. So here's an average of all the places we had Plantago erecta growing. We found was 13.4 grams of dry biomass. And then we measured the dry biomass in each of the recipient microsites. So here in this conspecific, it's 14, not too different than the average, of course. 15 grams in the congener site, really low in the confamilial site at just 4 grams, and then back up to something even greater at 16 grams in the site of its distant relatives. So we want to convert these measures into distance so we can compare them with phylogenetic distance. So we just take the differences here. And when these values in red are low, that means that the places we know the plant is growing are quite similar to the destination microsite. And when the values are high, like here, we're going into confamilial, that means there's a real difference in plant productivity. And we repeated that using soil moisture and soil salinity, which we measured using pretty standard protocols. What we found was that there was really no strong correlation between our niche distance metrics and phylogenetic distance. This was the best one we were able to uncover. Here, we've got phylogenetic distance on the X and productivity distance on the Y. I've removed the sites where phylogenetic distance is 0, or the conspecific sites, because that would just put a bunch of points down here when we would get this really strong leverage relationship. So I took that away, and we've got a correlation here, but it's weak and non-significant. And then I fit a smooth line there, and you can see maybe some threshold in here, but no strong evidence that phylogenetic distance is standing in for differences in productivity. And even less evidence that it is correlated with soil moisture distance or soil salinity distance. So I think what that says is that phylogenetic distance is really representing unmeasured niches, things we didn't measure. Either that or our measurements of the niche were really poor and do a poor job of describing the things the plant actually cares about. So just to wrap up, we found that relatedness does predict ecological similarity in site preferences, as well as competition, and that these patterns are strongest below the family level, so there's some kind of taxonomic or phylogenetic threshold above which it stops together and things become more variable. And phylogeny seems to be a decent proxy for ecologically relevant niche axes. And by that I mean the niche as our transplant experiments revealed. So the performance of our plants in these different sites represent whether or not that plant likes it there, says something about their niches. So phylogeny does a good job of explaining that. What those specific axes are either hard to identify, meaning we picked the wrong ones to measure, or they're hard to measure, meaning we measured them at the wrong time of year, or our methods were too coarse to really capture the dynamics that something as simple as phylogenetic distance was picking up on. So with that, I'd just like to acknowledge my collaborators and be happy to take any questions if I have time.
Hi, Gene.
So Brian's going to be around right after the session if you have questions, but we actually need to move